We talked about 3D comparison, pairwise alignments. Today we get into multiple alignment and the reach of comparative modeling. Science communication, don't forget about that. Ask questions. You can show structure in terms of 3D, 2D, 1D. 3D, 2D is equivalent, except for a mirror image. 1D, you reduce the information. 3D comparison creates families of related proteins. This is one particular superfamily, a large family. What you see here is a superposition of over 50 structures in this one family, and arguably they're very similar to each other. Dynamic programming gives you, dynamic programming is a way to completely do an optimal alignment on a scale of the complexity or time of the length of the two sequences. It's a simplification that works only because the major assumption here is that I and I plus 1 is independent, so, and you have a directionality in one direction. So you don't have repetitions in it that are essential. Or that's not a major issue. Anyway, so you speed it up by fast matching. BLAST matches words or not words. Words as defined by the BLOSUM matrix. And in between here you use another dynamic programming. In order to do that you need a good idea about the background. And now when you, in fact, did I show you this plot? Yes. So when you do an all against all comparison of 3D structures, you take the entire PDB, you use a sequence unique subset of the PDB, take it out, compare that as three-dimensional structures, everything against everything, or it's sequence unique against everything. Then you look at all the pairs that are clearly similar in 3D structure, all the pairs that are not similar in 3D structure, and in between it throw away. And then you look at the sequence alignments. So now you have pairs that are red, different structures, blue, same structure, and you ask, given a sequence alignment, dynamic programming, BLAST, or whatever you have, how well do I reproduce the separation into red and blue? And essentially you look at, upon intruding into the twilight zone, what the number is that we show here is the distance from this curve that looks at sequence length versus sequence identity. The curve is this one, the H is a P curve, or H-val. Plus 5 means above the curve, plus 10, so these are the steps, minus below the curve. And that, in fact, is the curve here. At some point above the curve, you have no mistakes. Red is 0, essentially 0, close to 0 here is 10. At that point where there's still a 0, you have, in this particular case, about 5,000 hits, positive hits, blue. Now, then the false positives increase dramatically. The true positives also increase by more than an order of magnitude, intruding into the twilight zone. And this is the entire aspect of the twilight zone. You have an explosion of negatives in the twilight zone. You just move a few percentage points from 0 mistakes here, 1, 2, 3, 4 points down, 50-50 mistakes, 4 down, 10% right. So within 8 percentage points, you go from no mistake to 90% mistakes. And that's a tremendously rapid transition, phase transition. If you think about the problem in terms of just not biology but letters, you can predict that there will be a phase transition and that there will ultimately be some rapid behavior somewhere. Where exactly it's placed, for that you need the assumption of biological sequences, and you in fact need to map it to the problem as here, structure matching. So blue is similar structure, red is not similar structure. Last semester some of you have seen the same sort of curves for similarity in function, protein interactions, subcellular localization. Those are different ones here. Anyway, now the incentive to intrude here comes from the fact that blue increases by more than an order of magnitude. So I actually want to intrude into the twilight zone because there's a lot to be found. The way we do that is by multiple alignments or by profile-based alignments. Okay, let's begin with the first step toward multiple alignment. Multiple being 3. So rather than aligning 2 sequences, you can essentially do the dynamic programming in 3 dimensions and align 3 sequences. Can you? You can. This is a little bit complicated in the dynamic programming in 3D. In principle, you can do it. You can actually hack yourself into a solution, and I haven't seen that solution, but somebody claimed that they had done it in a computer, to have a hack that uses 60 terabyte of main memory, and it lines up to 6 sequences. So is that realistic for a protein family, that you can align 6 sequences? So is only one person jumping up? One person jumped up. How many people are saying yes or no? Yeah. And? Is it realistic, 6? It's totally not realistic anymore. Utterly unrealistic. If you align human against uniprot, we have 20,000 proteins in human. How many of those would match to fewer than 6? I don't want you to give it the number 787, just a ballpark figure. Will it be 1,000? Will it be 10,000? Will it be 100? Where do you believe you lie on this?
None.
Pretty close, very, very close to reality. None plus minus 10. So the last time we checked, the number was in the ballpark of less than 20, fewer than 20, and that depends a little bit on how you do it. But none is a perfectly fine answer. So 6 really is by no means realistic. There is a hack that was published a while ago, essentially by David Lipman, that essentially does a dynamic programming pairwise, and in this vicinity of the intersection, you use a multiple alignment. That's a hack, but again, this hack is not really fully scaling. What's the typical hack that is used to align families of more than 6? Don't call the method. What's the idea behind the hack? Any idea?
Something like a consensus system?
Yes, ultimately what you do is you boil it down to a tree. Essentially you map it back to a problem that you can handle, which is a pairwise sequence alignment, and you do tons of pairwise sequence alignments. Introduced originally by Rost. And one way in which you can do it is the following here. So you begin with, in this particular case here, for sequence A, B, C, D. You run A against everything, and you establish, and B against everything, C, blah, blah, blah. And then you establish the highest similarity. So in this particular matrix that I take, it's chosen such that I can begin at different places. That means I can as well begin with A. The most similar one to A is B. Okay, so first, one way of doing this here, in step 1, I align A with B. And then I come up with a consensus sequence. Consensus sequence, okay, this example is badly chosen here. The consensus sequence in the example chosen is identical to the first. What I mean is, whenever you have the same sequence, the residue letter aligned, you take that letter. Whenever you have a different one, you take the one that is most informative. And it just, in my example, happens to always be the first one. Which means that the consensus sequence actually is you create a sequence that for most cases will neither be the first nor the second, and in particular it will not be an existing sequence, but a sequence. Okay? Then you do the same thing for the next most similar pair. The next most similar pair, A and B are aligned, so the next most similar pair then is, well, it's only C and D left in my example. So you do the same thing here, and then what do you do next?
Pairwise alignment of those two.
That was so trivial, and my questions are getting so trivial that nobody will answer anymore. The problem in these trivial questions is, thank you for your patience with me. It's difficult for me to see what is so trivial and when is it that I ask such a dumb trivial question, and when is it that I lose my audience. And it's just like a phase transition. One step more and I lost you, right? And I don't like that. I don't want to be alone in the room. So yes, that is one way of doing it. You could do it in a different way. You could begin with step 1 and say, you do this pair, and then in the next one you do the pair, or the consensus of these against the third. So building up the family in some sense. And there are many other ways in which you could do it. You could pick the seed differently, but in principle it's always the same hack. You reduce the problem, you map it onto a tree, you map it onto the problem of a tree of relations, not an evolutionary tree here. That may or may not imply an evolutionary tree, but ultimately you map it to pairwise comparisons. And it does not even have to be a tree. You could sort of have a random pairwise comparison. And if you want to spend a little bit more CPU time, you just look at all possible pairwise comparisons and all possible unifications, pairwise unifications of those. Okay, ultimately, what we try to do, we still try to actually build up something that is in the safe zone of alignment. So far, we are not building a refined search. If we could align two sequences and say they have a similar structure because they were sequence similar enough, then by the multiple alignment, as I described it so far, we're not really building up additional power yet. What is it that I need in order to build up the additional power? I do a little bit, and I'm overstating my case, but what is it that I need to sort of build advantage? Gain distinction power. In fact, building up my consensus sequence in this way, I already gained something. That is already better than just the pairwise alignments. So I have a gain. But what can I do that may be more accurate than the consensus sequences? That describes the group. I suspect this is really very, very obvious again. Is it? Or is that one of those moments where it's not that obvious? Does everybody know or not? No. Okay. What describes a family? What I mean is essentially profiles, right? PSSM or motifs. So Thierry's work gave these slides. These slides are not exactly for alignment, but ultimately the idea of building up a profile or the motif is shown here, where you have a transcription factor binding site, 0 here, then you have upstream, downstream, so you have minus and plus, and at every single position you have a probability of observing that particular letter. The higher the letter, the higher the probability of observing that letter there. So the cysteines here, and now you can take this observation and can turn it into a motif such as this one, a regular expression. Okay? The regular expression will immediately allow you to say which of these letters match. In fact, this particular is chosen such that they all match. Now, in order to build up PSSM, a position-specific profile, substitution matrix, or profile, or whatever else you need that, but it's specific, that is the protein-specific, you begin with a set of sequences that you pairwise align. For instance, in the way in which we did it before, in the hack of pairs of pairwise alignments. And then what you do is you simply count at every single position here, how often do you have each of these 4 letters. This is still MER count position 1 and so forth. And then you turn that into a relative frequency, you get a table that you do the log odds, and now you have for every position 1 to 6, for every one of the, in this particular case, nucleotides for letters, you have a log odd matrix that replaces the substitution or scoring matrix you started off with. So the 0 model essentially is the BLOSUM matrix. Before you know anything, that's what you look at, and then you build up the specific matrix that replaces this one. It replaces this one again by aligning along the sequence, and you fill in for every single amino acid of the 20 a probability, so to speak, of matching there. It's not quite the probability, it's a substitution matrix. The difference simply is that they're negative values, okay? You can translate that into a probability. So far, so clear. And you've seen that before, right? Again, this differs because now you essentially have a BLOSUM matrix, a different BLOSUM matrix for every single position in the protein. And that BLOSUM matrix is supposed to be specific for that family. While the generic BLOSUM matrix is the generic alignment matrix that measures biophysical similarities. these profiles profit from relations and families and build up also the substitution matrices. Now, what do you believe is then the difference between a profile and a motif? Or profile being, for me, the simple word to say position-specific scoring matrix. Today, people use mostly PSSM for that. What's the difference between a PSSM and a motif? Well, one way of formulating motif is the motif of the regular expression. In that way, if that were your answer, then the difference really is the probabilistic idea. The generic difference or typical difference that people say, typically people refer to as a motif as something that is shorter than a profile. A profile typically is the sort of thing that captures a domain-like region, while a motif typically is a binding site. Okay? But in principle, those are similar things depending on what you call a motif. If the motif is a probabilistic thing, then they're essentially similar. Okay? Now, the basic idea behind PsyBLAST is to, in fact, use that motif for the alignment. And again, the specific motif replaces the substitution matrix. The background, or the person behind it, is Stephen Altschul at the NIH, at CPI, who has relatively few publications, okay, 2 years ago, but the ones he did have an immense influence. And I would argue that with an H index of 44, this by no way measures his impact on the field. There's no, you know that PsyBLAST is popular. I'd argue there's no drug discovered. There's nothing ever done in experimental biology for people who use genes or proteins without beginning with the PsyBLAST. It's always at the beginning of everything. It has immense influence. Another person behind PsyBLAST is the director of NCBI, long-term director, David Lipman. In this particular case, the H index is a little bit higher. He made it happen. And then there is a whole team behind it, and this team, I said already the last time, includes Webb Miller. Now, the first step is the fast hashing. That's the same that we had before. In the next step here, the original BLAST extended in either direction. By the way, the first one is done already using substitution matrices. The words are already done on the substitution matrix. Then we have an extension in the newer BLAST. I mean, new is from '97. An extension beyond the immediate extension. You have an extension by running a dynamic programming. And we briefly discussed that already. How come that dynamic programming you cannot do for these two sequences you started with, but you can do a dynamic programming in between? What did you gain by finding the words? Could I have somebody say at least whether that is the trivial, you're not answering because it's trivial or because it's unclear? So again, dynamic programming I can do for two proteins, but I cannot do one protein against an entire database. Or can, but I need a lot of CPU for that. It's not fast enough. That's why BLAST was invented, okay? That's why we have a fast hashing. Now, I say that we do the fast hashing and then we are back to dynamic programming. Why did we do the whole thing if we're back to dynamic programming? What's the big gain?
Because the fragments are much shorter and so it's much faster.
The dynamic programming is an algorithm that is proportional to the length that we compare the square. Sorry, not to the length, to the square in the length. That's the main point here. So if we cut down the protein by a factor of half, so we have only a single motif somewhere in the middle, then we increase by a factor of 4, half by half. And that is the major power that we gain here, is because we square. By finding 5 fragments, we reduce by a factor of 25. And that's the reason why we suddenly can easily do the dynamic programming. That's the point here. We still need to get the statistics right, same as before, the background statistics. Now what we do is we collect all the pairs. So we run one sequence in this way against the intended database, build up the profile, then we turn this profile into a position-specific scoring matrix, and in the next run, we take the profile and run against the database. That's clear, right? Is it? That's the major thing. So now you replace, in the second step, you replace running a sequence against a sequence, essentially a pairwise alignment, by running a profile against a sequence against a profile, symmetric thing. Clear? And it's, again, usually you do that too. You do by pairwise alignment. You also do sequence against profile, except for it's the generic BLOSUM profile in some sense. Here's the position-specific profile. In terms of running the method, it's all the same. It's just the lookup at every single position here becomes a different result. Okay, that brings us to the last step, which is the trivial one. The I stands for the position-specific iteration. So we do it again and again. When do we stop? When I've eaten up all my CPU time. No, is there a better answer? This is one that is trivial, right? Or?
When the profile doesn't change anymore?
That's a great idea. In fact, that is what people typically don't do. So typically, we in fact, so, no. So let me step back here. What you have to understand is that the beauty of being in this field is that many of your ideas could really change the field to the better, turn the field into something better. In this particular case, unfortunately, the idea is not such that nobody ever had it before. The problem really is how to realize it. I believe it would be much better than what we do. And in fact, so in '98, our group tried to put in something, and in '98 it worked, but then PsyBlast changed something. And so the problem here is to capture the criterion of change. So change, is that in a bit? Obviously not. So change, somehow we want to measure important change of the profile. And there you get yourself into the problem. So typically the simpler way in doing that is, because I already have the information present without looking at any differential anymore, is if you do not add a new sequence. So in the last run you added nothing to the previous. So you have no new, so you run with the latest profile against the database, and whatever you find is the same thing that you found before. Then you stop. That's one criterion for convergence, and then there's the early cut. But what you said, Hauke, is really, it would be a better way if we knew how to do it. Okay, so let's just explain what is going to happen in a typical PsyBlast hit. So we begin with an unknown sequence. This, say, would be my real family that I knew, and as family I now define things that have a similar structure. Okay, my first hit, or my first search against the database is going to find some proteins out here, bless you. How, if I put some geometric, if I put some lines around you, right? How would they look? What geometric shape would I put around you to describe the first step in PsyBlast?
Very short lines?
Short lines. So it will be close to you. That's right. I'm asking now, my question is it going to be a square? Is it going to be a square with you being at the center of the square? Is that my search space? Is that my reach space of the first run, of the first step? Or is there something else that comes to your mind? A wiggled line, or a line that is somehow... Can anybody throw anything at me? I mean, names of lines, preferably. But you don't have tomatoes anyway. Coffee I would take too, so...
Circle?
Exactly, because you do pairwise alignments. And pairwise comparisons, it means that everything within some boundary, you put a threshold, whatever that is, an e-value, a percentage sequence identity, but it essentially defines a circle, with you at the center of that circle. That is the distance, right? So the ones you actually find may not be sampling that circle equally. So it may be that in the first round, the 5 hits you find happen to be on the right-hand side of the circle. What will that lead to? In fact, the probability that they equally distributed over the circle is very, very low. Most likely, any number, even if you had an alignment of thousands, there will be some preference with respect to the circle. So say the preference in that direction, because ultimately that may or may not be influenced by the shape of the family. But again, the probability is very low that it's really sampling the circle. What will it do in the next round?
It will shift the search space.
Exactly. That's the major point. Because since you define a profile now, you in fact no longer find the circle. Now you no longer look for a circle for the original one. And you sort of shift the zoom that you have. This area, what this egg is implying here, the area of the egg is larger than the blue. You see more through the specific profile, but it may be slightly shifted. Now, one aspect of that is that it could have been that there wouldn't be 5 but 50, and that in fact there was one protein in here. But this was one of 51, and in fact the focus was really there. In the second round, this one is lost. Okay, so the more refined, the wider angled psi blast loses something that the trivial pairwise blast finds. Does that ever happen in a psi blast search? When you look at the bottom of the psi blast search in the last round of profile, could it ever happen that you lost something that was trivial from the hits above? Yes? No?
Yes. I don't know. I just said yes.
Good.
No. No.
No. No.
Yes.
Because that's just a balance, because the first part of the table always has to say yes, except for you, you're the big exception. So in this particular case here, the majority was wrong. In fact, not only can it happen, these days that happens in every single search. And the trivial hits from earlier on are not repeated. So you have to actually go back to see whether you lost some of these simpler pairs. There's an important reality. And there are very good reasons for that. There's something to remember. So the way I built this up is sort of trivial. It comes from the power of the profile that you shift focus, right? And from the nature of sequence space, so to speak. You could argue that in order to do a format, you would repeat whatever you believe was the good hit at the end. That is a position. Another position is you want to put as few bits out as possible to reduce space. And that is the position that Cyblast, because of a very understandable reason, took. Just something to be aware of. Okay, now, while sequence space grows, another thing that I want to point out, we briefly talked about the sequence space hopping. The sequence space hopping is, we know in our first circle, assume we had this distance, the size of the circle that is such that we know that everything we put inside that circle has a similar 3D structure. We're absolutely sure. If that's the case, then we could use every one of these initial 5 points and draw a pairwise sequence search around each one of those. Start a new database search with every one of these 5. And then what we would get is this sort of light blue, which in this chosen example is bigger than what the profile search can do. And it could actually be true. Again, this is a potential that is not used here. It is, on a CPU level, something that is not easy, but it's something that people have done, and it's something that is not maintained on a large throughput search, but in particular cases you possibly could try something like that. Okay, let's go further. Let's do a couple of iterations. And let's say at some point of the iteration, you pull in a family that has a different structure. Mistake. Okay? Now my question to you is, what's going to be the next step? Say what you pull in here are 5 or 10% of the proteins. Will it matter? So I say, so far what you find is 1,000 sequences, and now you put in another 50. Will they matter? From the wrong family. And now you do another round with a red profile. That you know, in my construction, is not quite right. You know that the one before was better, but will it really matter?
I think you would always extend the profile such that it meets both families.
Yes.
So you would, yeah.
And?
Therefore you would find proteins from both families and use the original property.
But would you find them in the same ratio? 5% those?
Keep iterating.
No, in the next step.
No, in the next step you would preferably find proteins from the green family.
Who is with him on that? Who is not with him? So there's nobody against you, everybody else is sort of unsure. And only if I push them they will say something, or they will... so the point is, you're wrong on that one. The point really is that, imagine you had a profile, and in this profile you had 4 entirely conserved columns. And those, in fact, define that structure. There's something about that structure that really needs those. One wrong, you destroy the entire importance. One wrong, and you suddenly completely get away from what was essential. The central thing was that is not buried. It's be it 5%. But you open, in some sense, you open it much wider than 5%. Although you just add a probability, this is a completely misplaced probability. And by that you hit yourself. Because you build up your 1,000 sequences all pointing in the same direction. And that completely destroys that direction. So the moment you begin to pollute, you actually pollute more than just the number of sequences you pull in. Okay? So the beginning of pollution is really, really dangerous, and that is what you have to absolutely avoid. In the olden days, so when the first few years when PsyBlast was published, pollution was a regular thing, and people just did an early stopping. So rather than looking at does the profile change, they simply said, I do 3 steps, or I do until I have at least hits at 10⁻3, or I use a very, very careful threshold for the first round of, going into the iteration, I use 10⁻10 as a threshold, and then only in the end I do 10⁻3. The problem really is that this will decrease the presence in the room. Do we have a short break for bio break, water or coffee or something? The problem is that the ones who need it most are not with us to answer that question. Okay, so I wanted to avoid that. Okay, so we do a sequence against a profile, and I said that PsyBlast is one way of doing that. Is it the only way of doing that? Is it the only method that you know that does something like that? Or let me ask it differently. Do you believe PsyBlast invented that idea of doing a sequence against a profile? Can you name any other method that would do something like that? Rather than pairwise sequence comparison, they do a sequence against profile? Position-specific profile comparisons? Anybody in the room has heard any method that does that? Other than PsyBlast, yeah?
Depends on what you claim as a profile, because I think there are profiles which are more based on biochemical properties, and you can build a clustered biochemical property for one family, and then you have a biochemical profile, and you can compare a new sequence against this biochemical.
No, but that's exactly what you do in PsyBlast, right? Whatever you call it, you call it biochemical property specific to that family. You call it profile. You call it position-specific scoring matrix. You call it omega, or whatever you want to call it. The idea is the same.
But isn't it different when you also put in information about whether a residue is buried and all these things?
Ah,
that's what I meant.
So you replace the residue by an object that has other features.
Yeah.
Yes, but ultimately, okay, yes. Let's just stay, we will get to that point. Let's just stay on the letter feature environment. Any other method?
What about prosite? That's more these patterns. Or
Prosite is originally really evolved from the idea of regular expressions. So the first prosite motifs that Amos put in there were really regular expressions. It then evolved into something that is profile-like. Yes, that is true. But it began more as originally it was a more, today prosite is exactly that sort of thing. It is exactly like PFAM in principle. But originally it was not. But prosite is also not really an alignment method. I meant an alignment method. PsyBlast is an alignment method. Sequence alignment method. Okay. Have you ever heard of Clustal? Clustal is just one of many methods that did it long before PsyBlast. The big issue is that PsyBlast did it in a way that was fast and easier to handle for many. So Clustal, again, the first generation of Clustal was in '88, or the first Clustal the Clustal V method existed then. Now there's Clustal Omega. There's a bunch of co-authors. The basic idea is essentially the one learned from Doolittle. You do an all against, you begin with a family of things that you know are related. I could use a BLAST for that. Historically, that's exactly what they did, or FAST-A initially. Then you build up this matrix, you take this, from this matrix you build a tree, and then you align according to that tree. And that brings us to this issue of phylogeny. And let's just look at an example. So you duplicate that protein, be it a single domain protein, you duplicate it. Upon the duplication, you essentially create a two-domain protein. Since one domain already does what the original one did, you sort of over evolutionary timescales can possibly evolve this to do something else. Then you go further, you have another duplication now of two domains. The other one here does something else. And so you evolve an object that sort of differs from there. That's one domain history. Story number two is you start with this one, you begin to sort of cut because you see there's a duplication event, and you keep evolving and go into something else. Story number three is this one. Now the point is, what you see today is this. In what you see, you may find relations that have nothing to do with the way it actually evolved, because we don't have the entire history. What I drew in my toy example here, in the beginning, so from what you see, you would build this multiple alignment. From the truth, this is nowhere near the truth. This is not at all a recapitulation of that evolutionary event. And we do not have the evolutionary event. That is something that so trees are very intuitive. Trees lend themselves to describing the sequence alignment problem, because essentially we map it onto pairwise alignments, which is a tree structure. We use hashing, tree hashing and BLAST. So all over the place we have trees. So using a tree to describe evolution is just intuitive. But it's not really supported by observations. So that's something we have to, it's something that is very much of a model. Back to Clustal. It uses the tree, it does pairwise dynamic programming. One problem of doing all of that is it does a dynamic programming for everything. What's the problem? It's speed. And speed hits you when you go into large families. So now the Clustal Omega sort of can do some large families if you have enough CPU. But large families being 20,000, 30,000, 100 is in fact, some families now are over 100, and that is really beyond what Clustal Omega even can do at this point. Clustal is not only earlier than PsyBlast, it is also quoted quite frequently. As you see, here's a bunch of these publications. Just back to the people behind it, Des Higgins. It started all with Des, relatively high age index, but the major thing is really here. not many publications, but 2 over 90,000. The other person in there is Toby Gibson. So Des was the one who is the programmer. Toby is the one who looks at the dynamic sequences, at multiple alignments. He's the expert. He just looks at 2 proteins aligned and tells you whether it's right or wrong. And he can steer it at 50 and 100, and he's still amazingly accurate in guessing that. So the team, Toby and Des was fantastic to develop Clustal in particular, because now what you can do is for every single pair comparison, you can use a different substitution matrix. You can use different weights or different penalties. You can do that according to the level of similarity you observe. You can do that according to other features. And that essentially is what Toby allowed to optimize. The one aspect that is missing is the visualization that came from Julie Thompson. And ultimately, I'd argue that it's this team of 3 that made Clustal so successful, because you can just put the Clustal figure into your paper. It's easy to do. It's very, very simple to see. And you cannot do that with PsyBlast. Ultimately, what people use Clustal W for is to create a publication figure. They begin with PsyBlast, run the Clustal W, get a nice figure. And that, I believe, is the reason why this alliance of different methods here is so successful. Maxum, the major point, so the people behind it are Reinhard Schneider and Chris Hunter. The major point in the difference to what we had before is that we now have a sort of, in principle it builds up like Clustal W, a progressive alignment, so it does AB, AC, AD, and then builds up conservation weights and profiles systematically, but it has a protein-centric view, where Clustal tries to align an entire family. Methods such as Maxum try to align a protein with the rest of the universe. So one aspect where that is different is in the protein that you care for, that you start with, you do not allow insertions. It's the protein for which you want to know all the binding sites of the others. The protein for which you want to know how it binds. This protein is a real object. It doesn't have an insertion. Okay? And that is somehow applying Clustal or Maxum. This is something you have to take into account. Again, Clustal is more interesting when you think about evolutionary studies. Many people use Clustal because it's the nice figure. But actually, most of the people who use Clustal use it because of the wrong reasons. Most of the people, experimentalists, what they care for really is the alignment of one protein. That is the one described in that paper against the universe. And Clustal is not the best way of doing that. It's just that the methods such as Maxum are not around and don't make nice figures. Another way of doing the math is hidden Markov model. We've all heard of the hidden Markov model. So what's the feature of a hidden Markov model? So one way of putting the hidden Markov model is essentially you have hidden state 1, you have a transition to itself, you have a transition to the next state, and then transition, in this particular case, to the end state already. You have an observed sequence of symbols, and you map the observed sequence of symbols onto that model. So it's a probabilistic model. That is one important characteristic. But what's the reason for the word Markov?
It doesn't have memory. It only depends on the previous state.
Yeah, which is some memory, right? Is a memory, right? If I remember where I come from, I don't know about you. Sometimes I don't have that memory. Yeah, you know, when you're in that moment, it's not. Anyway, not really funny. But so we have one-step memory. Okay, you know, t minus 1. So the decision at the time point t is dependent only on t minus 1. The alignment on position I plus 1 is only dependent on I. So you transit depending on what the state is here. Now, so the slides come from Marco Punta, who now is not in CNRS Paris anymore. He's now in the Cancer Center in London. And Marco likes to play football. He likes coconuts, but he also likes to play football. And he picked out this, when he was still in Germany, he picked out some results from the German soccer league, Bundesliga. And one particular team, Augsburg, played against all the other teams. Augsburg lost is red, Oxboard 1 is blue, and draw is in black. Now what you can do is you can simply go through the sequence and see whether you can predict the win, draw, or lose. You essentially have 3 outcomes. And given that today they won, what's the probability of either winning again, or drawing, or losing in the next game? And how would you do that? And then 0 level estimate or prediction or model. How would you initialize the transition probabilities here? It's very simple.
You can just count like the 3 they won, so 3 times they lost, 2 times they draw.
Exactly. You look at the overall numbers, and then you simply take how often did they lose, what's the fraction of lose, and you simply look at the entire table. This is the entire table, and you compile you compile the transition. Now, what else could you use as information? I don't know. So the answer now, you need to. How are we about? Is that okay? If I open the window?
Yeah.
So now you need to learn a little bit about football. A little bit about football, about sports events, I believe, ultimately. Is there anything else you could put? Yeah.
Yeah, it could. You can use if it's a home game or away game.
Exactly. So you essentially can. Oops. Oh. Here's the criterion. So you can classify them by home away, and you can call home away are essentially hidden variables. Those are descriptors that you help you to increase the odds of predicting the outcome. And now you essentially use that as an additional criterion to change the transition probability. And here we have a simple hidden Markov model to predict an outcome of this, right? So we have discrete alphabets, probability, emission probability, a transition. It's a simple model. Now what about this one here? We have 3 states. Is that the same? The traffic light. And what I assume here is the traffic light that we all encounter. So it's not sort of a fake traffic light or something. It's a real traffic light. Is that a hidden Markov model? No, it's not. Why not? Somebody.
There is no probability.
Exactly.
It should be
so totally deterministic. That's what I meant, provided we have a regular traffic light. So we don't have some electrical problem or something like that. Or the guy who programmed it was not colorblind or something. What about this one here? So in this particular case, what we have sun, cloud, rain. Is that a deterministic model that we could map onto a hidden Markov model? Anybody feels not? Anybody argues why not? Just because it's a tough task is not good enough. So yes, you can clearly translate that into a hidden Markov model of this sort of type. And this leads to a set of hidden Markov models. The major ones are currently in use are SAM and Hammer. And by the way, by now it's, I believe, Hammer, or Jackhammer now, that goes back to Sean Eddy. The idea of bringing HMMs into biology came from Anna Schroch, who visited with David Hausler at UCS Santa Cruz, SC, from Copenhagen. And he was fascinated by the Markov models and convinced David to begin working on sequence alignments. So he was a young student at the time who thought that hidden Markov model is a cool thing out there. He had worked on neural networks before, and now felt that hidden Markov model is about, he went there in '92. And by the way, he went there mostly because California is sunny. So just fun, and he brought fun to a fun guy, and then Kevin Karplus somehow took over the design of a generation of hidden Markov alignments. In hidden Markov alignments, simply that is your model. You have a match state. So in this particular case here, you would have 6 residues, 4 residues. You can match or not match, and you have an insertion and deletion state. Let's not do this. Let's just begin by building up the slides. I don't like the slides that come, and we're just going to do it on this slide. And then I'm going to zip through for the next 5 or 10 slides. So how do you do that? The first question is that a good example for a model for a pairwise alignment? Can you do, with a device like this, it's a probabilistic model, can you do pairwise alignments? So again, the alignment is done by defining the transition probabilities, or by defining the individual deletion insertion penalties. If you had to align 2 sequences, and you knew nothing other than these 2 sequences, and well, whatever, you have a computer and you have the internet, could you fill up these transition probabilities? Yeah, sure you can. You use the BLOSUM matrix, right? That's the simplest way. You take BLOSUM, translate it into probability, and then this may not be this is clearly not the smartest way of using a Markov model, but that's the first step. Okay? You do pairwise alignment. In fact, you initialize. It's not a search method here. It is a method that aligns a family, just like Clustal. I believe I didn't make that point clear. I said that Clustal takes a lot of time. That means it's very, very difficult for sequences of 100,000 or families of 100,000. But that assumes that somebody gives you the 100,000. This is not Clustal against the entire 60 million. That is not really. So typically what happens is that people run a PsyBlast and then Clustal W. It's the same thing here. You have a family. But now what you want to do is to do the alignment of the family better than PsyBlast can do it. But it's somehow considering the entire family, and that you can do. But the first step then is you take the first pair, right? Where do you get the transition, the deletion and insertion parameters from?
The gap penalties.
Yes, you take it out of the air. That's the first step. You take the one that you usually use for dynamic programming. In Clustal or wherever, you take the first choice. And then what you do when the next one comes in? You align it according to that, and then you change it according to what you then produced. Now you have 3 aligned, and that gives you a 3-mer model, and that sort of re-estimates the properties. And that is essentially one way to imagine. All the slides that I'm going to show you completely contradict this. But this is not what you do in practice. But this is the idea of it. Okay, and that I feel is more important. In this lecture, I don't take enough time to explain to you how the Markov model is really doing the alignment. For me, the important thing is that you understand the idea behind it. Now, here is the story with a sort of entropy kind of model. Where you start, then you build up according to minimizing the entropy. The entropy essentially changes by gaining information. The information you gain by looking what you find at every single step, by adding pairs of sequences, and you always add such that you gain information. Now, when you have the final model, that model is specific to one particular protein family. That's why PFAM uses them. PFAM is a database of protein families, and every single protein family is corresponding to one model. Okay? How is that now differing from PsyBlast? Now you come with a new sequence, and you align into that model. And you simply, the solution how to align that model is you find the most likely path. The transition probability, the insertion addition gives you a probability with which you multiply, and then you simply get the one solution that is the optimal given the probability. As simple as that. And in PsyBlast, what you would do is you would build up the profile in an iterative approach. You would freeze the profile, new sequence comes in, you would run it against the sequence and say if it fits or not. What's the difference between the two? So at that point, the difference really is the model. It's the way you compile probability. It's the way you use the information. That's all. Conceptually, it's the same. Since the hidden Markov model has had the opportunity to build up an optimal family profile, it has an advantage to sort of describe the family of a PsyBlast that sort of still built up an optimal pairwise set of pairwise profiles. So that is why I assume this is the best solution for, in the context of building a database such as PFAM, while the triggering of PFAM running in principle is still done by PsyBlast, or for many. Now we can think about something new, genetic algorithm. And the one thing that all the methods I talked about, dynamic programming, PsyBlast, SAM, they all agree on one point. They completely ignore the dependence of column I and J. Okay, so they assume that what you align at position J is completely independent of what you had aligned at position I. Is that biologically correct? Why not? It's biologically not correct. Can you give me an example for why not?
Correlated mutation.
That is true. So, other than correlated mutation, much on a, it's very, very related. Your answer is right, but there's something else missing. Or the actual reason is not correlated mutation.
The residues that are close and pre-structured are.
That's the actual selector. And that's why we see correlated mutations. So the logic goes the other way around. Yes, and you were smiling because you saw that on the slide, or?
No.
Yeah, because you are biased. Okay, fair enough, I forgot about that. Yes, so when you take the sequence, you put it into solvent, and it adopts a unique three-dimensional structure. You can only do that by long-range interactions. Long-range interactions is more than I in I plus 1 or I plus 3. So proteins, in order to adopt a minimal volume, have to form long-range contacts. Long-range contacts, if they are relevant for the formation of the structure, and I told you that the way the structure looks is always important for the function, then you will maintain these long-range contacts, and immediately you have a correlation that completely violates what all alignment methods do. Why do the alignment methods still work? Actually, we don't quite understand. So I could give you all kinds of explanations here, but it's all nonsense. The degree to which this should matter, we cannot estimate. And as long as we cannot estimate the degree to which this matters, the fact that alignment methods work is just empirical. And explanations for empirical observations don't help us. One way to get rid of that is the genetic algorithm. That can look at segments. So when you do Wikipedia on genetic algorithm, you find this antenna here, that if you look at it, immediately shows you how a genetic algorithm optimized this for capturing particular radiation, right? So nobody in this room could have ever done something like that by chance, playing around with a wire. I don't know. For me, it looks just what I would do, sort of playing around with a wire. But anyway, so this is an important thing that is optimized by genetic algorithm. The basic idea is you start somewhere, you have an initialization of a population, you compute the fitness of the population, and then maybe that is already enough. You go into a stop end, or you do a roulette on parents, and or do crossover on children. You mutate children, and then again compute fitness. Ask yourself whether you have reached the stop criterion, if so, and otherwise you mutate again. In visual way of doing that is shown here. For an alignment method, that idea comes into tea coffee, and again, of course, there's a reason for calling tea coffee, from Cedric Notre Dame, in the group of Des Higgins, who in fact, as you remember, is Clustal W's author, and Herringer. This is, by the way, if you want to do a postdoc or a thesis with Cedric, this is really in front of his lab, his office. That's the view, that is Barcelona, and that's the Mediterranean Sea. This is the beach, that is a public beach, and it's really, really nice. So it's not only for a special event, that's Cedric. Okay, the way you do it, you begin with a library of local, for instance, local and global alignments. Or you could begin with a library of different gap penalties. Or you could begin with a library that is created of alignments produced by different BLOSUM matrices. That's the input. Okay? And then, essentially, this is the genetic algorithm. Then you optimize, you iterate through this, and you get an improved multiple alignment. The beauty of that, what was the funny one this time?
It's because the guy who named it tea coffee put in E in alignment to get a tea coffee. So the abbreviation. And that's why I was always writing wrong.
Yeah, I can get to that point in a minute. Anyway, so I'll tell you why it's called tea coffee. So the big thing, so local global alignment improves, and then multiple alignment is the optimal solution. But the big thing really is you have now a way to code contacts. So to say that you have a system bridge, or to say that there are particular constraints you have on the 3D structure that are contacts, and you put them into the alignment. And the language in this formalism of the genetic algorithm, that's easy to do. And the language of PsyBlast, Clustal W, or HMM, it's impossible to do other than through a hack. A hack could be you force these two to be together. Otherwise you don't allow it. Here you just give a probability for this to survive. Okay, you increase the fitness. You allow things that violate the constraint, and maybe that is the solution you will find, but the one that maintains the constraint may just add it to your fitness. And then different optimal criteria will survive in the end. And the other thing is you don't necessarily have to define a certain set of those. Anything you know will help you to do the alignment. That's the beauty of the approach. And it's fantastic. So why is not everybody doing tea coffee? Do you believe? Really is a fantastic formalism. Why is not everybody doing it? Well, you all know, I already said that many people use Clustal, many people use tea coffee, use PsyBlast. Why do you believe? What's the problem of this approach? CPU time. Sorry.
Maybe you.
I'm sorry. I didn't. It was too. I'm too slow. So this is just it takes an immense amount of time. That is the only downside to it. But it's so immense that it's a major downside. There are many others, and I just named a few here. But still, most of the people use hidden Markov model, Clustal W, and PsyBlast. Maxim I only brought to show the different philosophy. If you have additional knowledge about an alignment, this is a very good path to go, and if your family is not big enough. So what sequence does is goes sort of with the trivial zone, sequence profile goes furthermore, and this I don't understand. Is there anything more fancy that you can imagine?
Maybe profile.
Exactly. So in some senses the simple extension here, pairwise, multiple, sequence profile. So then what you would do is, instead of comparing a sequence to this profile, you would simply compare the two profiles vector by vector. Simplest way to do that is you compile the vector product. So for every single column, you multiply it with that column that you see here. Is that simple? Why not? So I set you up in some sense. You correctly answered is not simple because the way I asked the question is obvious, what I want. But why? What's the problem? The vector product is totally trivial, right? The non-so-trivial part is you can do the vector product in different ways. You can look at the edit distance, you can look at the Hamming distance, you can, and the real problem is that none of these ways is better than the other. There is no obvious solution that is right. And that is in fact the reason why sequence profile to be used by many people took a long time, because it's more complex than sequence. And profile is another level of complexity in some sense, and it took a very long time to really get through. There are too many free parameters and it's complicated to do. Another issue is it may be unwanted. And there are two things that I want to convene with this slide. The first one, just looking at this vector here, now you would multiply that vector, say you took the Hamming distance, multiply that vector with the corresponding vector in another profile. What type of information do you see here that you would lose? Or do you see implied in that slide?
Yeah, again, the
yes. Exactly. So in some sense, again, let me say, Michael, I set you up again. What you see is not really the correlated mutation, but this is exactly the way we explain correlated mutation, because what you see is a correlation. So the correlation would be that you have the R here in this sort of subfamily, then you maintain the positive charge, but there is another subfamily, and then you actually go into the tryptophan. So you drop the charge, but you keep something else in that site. So you would immediately suspect that you actually have three subfamilies here. Okay? And that information about the subfamilies you would lose, because you would essentially treat the vector either in the Hamming distance where you just see is the first one in R or not, or you would somehow replace it by the profile where you essentially would see 3R, 3K, and 2W. But you would not see that they are correlated. And not correlated in that sense, but correlated between parts of the subfamilies, right? That's another thing you would lose. And ultimately, another thing that you may lose is maybe that this part of the correlation is important for some aspects, and this, in fact, is a different structure. And by putting them together, you would actually not really see the more generic structure, but you would see two different things. So these are two different aspects of the same slide here. But ultimately the point is you can, in fact, lose something. Now, I'm going to introduce to you a sheep hack. How can you possibly do a sheep hack to profile comparison? And the sheep hack comes from Darek Shubilsky, who's now in Boston at the Broad Institute. And in terms of CPU, sheep hack means, so here's profile sequence, here's profile. You want to somehow find a path in between. How could you do that? Any idea how you could do somehow hack or use something that we already showed on the whiteboard today and use that for a poor man's profile alignment?
Consensus sequences.
How do you know?
It's obvious. I mean, a consensus sequence sort of is a summary of a profile, right?
Okay, we get back to that later. Sometimes it's not good to be a teacher. So we run the sequence here, put the probability or the position-specific profile, and then now we build up the consensus sequence by simply looking at what is the most informative residue and replace the profile by that sequence. Once we have that, we can now, instead of doing profile alignments, we simply do consensus sequence against profile. And we do the same thing that PsyBLAST has been doing since now almost over 20 years. So, and it finds a simple way between, no decision is to be made here on vector comparisons. We do sequence profile as always. It's just a new type of sequence. With a new type of sequence, we have to evolve a new statistics. There's a lot of detail in that one. But ultimately what we get at is, this is pairwise BLAST, this is PsyBLAST, so that's the increase. And with the consensus sequence, we get not as much more, but a substantial part. Today, mostly the only profile that really is used by many people is HH Blitz, that comes from Johannes Söding, who was at the LMU for some time and is now at the Max Planck Institute in Göttingen. And he initially developed that in Tübingen along with Andrei Lupas. That sort of sets up the pipeline of doing it, the fast pipeline, those are some statistics. The major thing, oh, I didn't want to get into that either. So the major idea behind HH Blitz, the way it does it, is in a similar way that is the consensus sequence, except for it replaces sequence by biophysical features. So it replaces a string by a set of features. In order to do the final thing, instead of 20 amino acids on a consensus motif, they do it on a 200-letter alphabet that uses essentially a consensus for a 200-letter alphabet. Same idea, same hack. And again, since this is 200 and there are all kinds of other features involved, getting the statistics right is highly non-trivial anymore. But in principle, the idea is, as Gregor said, obvious. That is arguably the only thing that really works in a high throughput manner for proper comparison. Not only works, but that is sort of easy to use, relatively easy to use. Easy to use, everybody can use it. And with these profile comparisons, we intrude deep into the twilight zone, into the midnight zone. So what is the midnight zone? Let's just look at this zone here. What we are going to do now is we're going to take the entire protein database, well, take some sequence unique subset thereof of it, and do a sequence unique subset against the entire database, 3D comparison, and look at all the proteins that we call 3D similar. Okay? Everything else we throw away. And what I'm going to ask you is, how do you imagine if I now read off the 3D alignment, I read pairwise sequence identity. And I ask how many proteins of the same similar 3D structure do I find that have 100% sequence identity, 19 da 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 da, right? The answer is this. 100, and then you have somehow here you have a peak. That gray, by the way, is the random superposition. What do you expect? This is not the answer, this is the expectation. Would you move anything in this curve? Again, maybe this higher, maybe this lower, maybe this there, maybe this there. I don't know. What would you move? Maybe this peak up. Any move, or is that sort of what you expect? Would you expect some bumps somewhere? And this is for the actual PDB, not for what the PDB would be if we knew our structures. Today we know 100,000 structures. So what I'm going to show you is an older version of it, but I assume it's somehow similar. Nothing?
Maybe a peak at 100, or at least a slide.
So more like this. Okay, here we are. This is the solution, and everybody's happy about that, right? The random peak. This is not true. It's absolutely not true. The real data is here. And this data is slightly fake in the way it's compiled. I had to sort of distinguish that side and that side to the extent that the number that you see here should be much lower than it actually is. So I way overestimate the counts here. That is where the distribution sits. That is random. That is real. The difference somehow, random is roughly 5%. 20 amino acids, 100 by 20, right? Roughly. There is a difference here. It's not 5. That difference is very, very important because we have millions of points here. It matters absolutely, and that in fact has to do with correlations with the fact that amino acids, so you cannot, I and I plus 1, you cannot have all combinations. As simple as that. There's a dipeptide bond, or there's a preferential dipeptide frequency. And that essentially is most of what you see here. The difference between these two, so if that were the answer, then we would have one structure. The difference somehow codes for all the difference between many, many, many structures and one. Somehow this is the difference here is the entire play space in some sense for the evolution of difference in structures. And that's a remarkably small number if you think about it. In fact, Now, what it also means, so this is, let's call them the anchor residues, 3 to 4% of all the residues are anchor residues. Now, the question is, could we come from that side? So you have two proteins that have absolutely no similarity in structure, and that suddenly evolve a similar structure. Well, since the peak is so close to random, that suddenly becomes an event that could have happened, right? It clearly means something else. 4 billion years of evolution somehow have reached equilibrium, because the actual structures cannot move much more there. They cannot be random. Okay, maybe they could have moved like a little percentage points. 1 percentage point, but there is not much move space here. You also have to remember that in the difference we also have to bury all the functionality. So this is already very, 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 very close to random. So most likely this implies that we had essentially enough time to try. What I mean here by convergent and divergent evolution is misleading. Convergent and convergent to the same structure. Divergent from the same structure is not, it's a misnomer. But the midnight zone here really is one that we cannot reach. And I believe I call it a day here.